All right, hello everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zimanyi. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. And here with me today is my colleague at NCCAT, Ed Ang. Joining from the Stanford SLAC cryo-EM Center S2C2 is Michael Schmidt. And here from the Pacific Northwest cryo-EM Center PNCC, the group who invited today's speaker are Omar Devloku and Lauren Vega. This webinar is a regular event that we hold usually the last Thursday of every month, although notably this month we have a bit of an exception because next week we will be busy preparing for an in-person workshop at PNCC. That's part of the American Crystallographic Association annual meeting. I'll say a bit more about that in a second. But already on our calendar for next month, August 25th is our next talk. We're still working out the final details with our speaker, but save the date now if you'd like to join us again next month. The talks in this series highlight the methods that groups are using to obtain and interpret the data they can collect with the help of the National Cryo-EM Center resources. And we hope these talks are useful, especially for groups just starting to use cryo-EM techniques. Omar is going to introduce today's speaker, Soumya Joseph, who's here from the Campbell Lab at University of Iowa. And he'll do that after the colleagues, my colleagues from each of the centers give a quick update on the resources that we make available at no cost to the research community. We're recording today's talk, and you can find that recording along with those from our past talks on our YouTube page or on the events page at cryoemcenters.org. For Zoom logistics, we're going to be watching the Q&A box for questions. So please use that and not the chat. And Omar will be moderating questions at the end of the talk. And today I just have 2 additional announcements before we get started. So if any of you are planning on traveling to Portland in the next week or week and a half, depending on which meeting, you can come see us in person. We will be at the ACA as well as microscopy and microanalysis annual meetings. If the word crystallography is confusing you, I'll point out that ACA recently introduced the tagline, the Structural Science Society, to reflect the many members that study structural science using methods other than crystallography, and that includes cryo-EM. And there are multiple workshops and sessions at the meeting on emerging cryo-EM techniques, applications of cryo-EM, as well as training. On Friday, we're hosting a hands-on practical workshop, actually on site at PNCC. We have a booth at the exhibitor show, booth 107. Also, I'll be giving a talk on Monday in a session called Educating the New Generation of Users, and have a poster that afternoon. So come and find us there. At the microscopy and microanalysis meeting, we'll be in the exhibitor hall at booth 1046. And that'll be right next to a booth from our colleagues at the National Cryo-Electron Tomography Centers, if you want to find out more about cryo-ET. And you'll also find NCCAT staff at the Monday poster session, PNCC staff at the Wednesday poster session, and Rosemary Haynes from PNCC is giving a talk in the Technical Advances in Cryo-EM session on Thursday morning. So we hope to meet a lot of you there. Come talk to us, and we look forward to seeing some people in person in Portland. One last bit of business. At the end of the year, we plan to, instead of a traditional talk, host a panel discussion. And we're wondering what topics are of most interest to our audience. So after our center representatives give their quick update, I'm going to launch this poll, but give you a second to think about it. We've thought some interesting topics might be specimen preparation, data collection, data processing, perhaps even just logistics of how to use the national centers. If you have other ideas, go ahead and chime in the chat. Or if actually you're not interested in cryo-EM at all, but really just want to hear about the science. That's also useful information for us to know. So chime in if that's the case for you. But I'll launch this poll in just a minute. But first, I'm going to turn over to Ed to tell you about what's going on at NCCAT. Ed, take it away.
Hey, I'm here in New York at NCCAT. We offer high-end instrumentation access to 4 dedicated Krios instruments. Our G4 Krios was just upgraded with a cold-effect and selectress energy filter. It'll be back in circulation this summer. Also, we're getting a Falcon 4i upgrade. In terms of our other categories of access, we offer chameleon access for blot-free vitrification, a robust cross-training program, and a very popular grid preparation and screening program. We welcome everyone to come on site. Last week, we had 11 visitors across our national centers here between NCCAT and In Situ, and we hope you come and see us soon.
Thank you, Ed. And next, I'll turn over to Mike. Mike, it's all yours.
Hi, I'm Mike Schmidt, and I'm at the Stanford SLAC cryo-EM Center, and also the Stanford SLAC cryo-ET Specimen Preparation Center, one of the centers that does tomography specimen preparation. We have four Krioses. One will be installed later this summer, and we have access on partially access on another Krios and a Talos for screening. We've had several people on site now transitioning from crystallography and other structural sciences to cryo-EM, and so we've had 3 sabbatical visitors for months at Stanford SLAC. We welcome all people from all professional levels to come use our facility and get trained and learn about cryo-EM and cryo-ET. So there's our website for the Stanford SLAC Cryo-EM Center, and we hope to see you here soon.
Thank you, Mike. And now I will turn over to Omar.
Thank you, Christina. I'm Omar Deboku, and I'm one of the SPOCs, or Scientific Points of Contact, here at the Pacific Northwest Cryo-EM Center. PNCC offers its users a seamless workflow from sample analysis and optimization to grid prep, screening, and automated micrograph collection, image analysis, and data processing to 3D reconstructions. We've also recently launched a screening access proposal mechanism that allows for users to come on site and work with us hand in hand, going from sample prep potentially all the way to data collection. In terms of hardware, we have 4 Titan Krioses, 1 Arctica, and time on a shared Glacios. All of our Titans are equipped with K3 direct detectors, and the majority are equipped with fringe-free imaging and bioquantum or biocontinuum energy filters. We are most excited about a recent upgrade to one of our microscopes, which now has both a Falcon 4i and a cold-fig. So in addition to all of that, we also offer many training opportunities, both remotely and in person, on topics ranging from sample prep, introductions to serial EM, screening, data collection, and data processing. I would encourage anyone with questions to reach out either here through the Q&A or at our website that you can see in front of you.
Thank you, Omar. And before I turn over back to you to introduce the speaker, I just launched the poll that I mentioned earlier. So we really just want to gauge what kind of topics people are most interested in, and based on what's most popular, what we'll do is pull together a group of both internal staff at the centers as well as some of our users, and just sort of collect their experience to understand how people are solving the problems they're having in cryo-EM. So I'm getting lots of responses already. So we'll let this go for another 30 seconds or so. And I think you can choose more than one option. So if you're interested in lots of things, go ahead and indicate that. And I think, let me stop sharing. All right, I'm gonna set a 10-second timer on the poll. So if you're indecisive, pick lots of things. and we'll let you know. So thank you very much for letting us know what it is you're interested in. And I'll go ahead and end the poll and turn over back to Omar. Omar, go ahead.
So it's a joy to introduce today's speaker, Soumya Joseph, who in the 9th grade, after being introduced to Mendelian genetics, knew that a career in biology was for her. From there, she went on to earn her doctorate at the University of Sydney in Australia. And currently, she's a postdoctoral researcher in Kevin Campbell's lab at the University of Iowa, where she uses structural and biochemical methods to study a glycosyltransferase involved in muscular dystrophy, and is currently supported by both the HHMI and Wellstone Muscular Dystrophy Research Network. Today, she'll be telling us about some work that she's done both here at PNCC and at APS, which includes her very first cryo-EM reconstruction ever. So Soumya, it's great to have you here, and please take it away.
Thanks for that introduction, Omar. Oh, can you hear me?
Yeah.
Great. Yeah, so this work on large one wouldn't have been possible unless I had facilities, national facilities like PNCC and national facilities like Argonne National Lab. So I'm going to tell you about how we think matriglycan is polymerized by a glycosyltransferase called large one. So large one is a single-pass transmembrane protein that is resident in the Golgi. It has a xylose transferase domain and a glucuronic acid transferase domain. And it synthesizes a linear polymer of alternating xylose and glucuronic acid. So the xylose here is the orange star, and the glucuronic acid is this half blue, half white diamond. And this polymer is called matriglycan. And it synthesizes the polymer of matriglycan uniquely on another protein called dystroglycan. This is part of the dystrophin glycoprotein complex. So people may have heard of Duchenne muscular dystrophy or Becker muscular dystrophy, and the dystrophin glycoprotein complex links the inside of the cell, the cytoskeleton, to the outside of the cell, the extracellular matrix. So in the absence of matriglycan, or in some cases where the matriglycan is very short, we get something called glycosylation-deficient muscular dystrophies. And these can be very severe. They can be congenital muscular dystrophies, or they can be quite mild, depending on the mutation. And it can just be like limb-girdle muscular dystrophy, that's very late onset. So here is a Western blot of matriglycan. So we have anti-matriglycan antibodies put on this Western blot. This is the matriglycan staining for a normal, healthy person control. And this is the matriglycan staining for somebody who has a congenital muscular dystrophy. And you can see that the band is less intense and it doesn't quite extend up to here. So the matriglycan, we believe, is shorter. These are muscle sections from a healthy control. This beta dystroglycan is just the core protein without the matriglycan modification. And here you can see we've stained it with the anti-matriglycan antibody, and it lights up. For the patient, the core protein is still there, but the matriglycan modification is very attenuated. And you can see in the H&E, this is kind of diseased muscle, what it looks like, and healthy muscle cross-sections look like this. So mutations in any of the glycosyltransferases that polymerize any of the precursor stem sugars for matriglycan synthesis, or large one itself, which polymerizes matriglycan, can cause these secondary dystroglycanopathies. So because matriglycan length matters, we wanted to know what the structure and the mechanism of the enzyme that polymerizes matriglycan is. So the enzyme that polymerizes matriglycan is large 1. And essentially, we want to generate and test various hypotheses on mechanism. So large 1 is, like the name says, a bit large. It's 200 kilodaltons in solution, so it's a bit big for conventional NMR. We and others have tried crystallizing it. We weren't able to crystallize it, but we were able to use single particle cryo-EM to determine the structure of large 1. So some of the questions that we asked was how does large 1 know the length of matriglycan to make? Clearly the short one isn't enough. And do the tandem at xylose and glucuronic acid transferase domain predicate processivity? So large 1 is expressed at very low concentrations in the Golgi apparatus. It's so low such that we cannot actually see it on a Western blot. So the only option that we had in order to reconstruct a volume was to make a recombinant construct of large 1. And because it only has a single transmembrane domain, we thought we could just, delete or replace that transmembrane domain with a flag tag, and then it's already in the secretory pathway. So what will happen is it'll just get secreted into the cell medium. And so we transfected a plasmid encoding large 1 delta TM into HEC293F cells. And these cells, they were cultured in suspension in a bioreactor. And essentially, we just purified the soluble recombinant protein from the cell media. And so this, prevents any effects from toxicity. We can get a huge amount of protein, as I'll show you in the next slide. And then we go ahead and validate our construct for both enzyme activity, monodispersity, prior to trying any cryo-electron microscopy. So this is a purification gel of large 1. This is actually a Kunasi Brilliant Blue stained gel. This is what you get in the cell culture media. There are all of these secreted proteins, and this giant blob is large 1. It expresses it has some of the best expression compared to some of the other proteins I work with. Some of the other proteins you can't see anything, and even if you try and do nickel NTA purification or cobalt resin purification, there's hardly anything. So this is the purification from on cobalt resin. One of the things we did to confirm that the enzyme is actually well folded and active was enzymatic assay. So here we have a 4-methyl umbelliferone group connected to glucuronic acid and xylose. This is fluorescent, and so we can introduce this moiety and UDP sugars, so in this case UDP glucuronic acid, do the reaction in vitro with the recombinant large 1 and run it on a reverse phase HPLC. So this peak corresponds to the reagent, and this peak corresponds to the product. So now we can test whether our recombinant protein is in fact active. We can also do the same experiment for xylose transfer, and in the case for xylose transfer, this is the product and the reagent elute earlier. So we know we can make plenty of large 1, and I'll tell you this is such a blessing because you can do whatever experiments. Protein is never limiting, whatever you want, and it's such a blessing not to have to transduce cells with viruses every single time you want to express protein. So this cell line, because it stably expresses, we just passage it in perpetuity, and we've been passaging it since I would say 2017 until now, like the exact same bioreactor. And we harvest from the bioreactor every week, so we are never short of our protein. So going over the domain structure of large 1, as I said, large 1 has a transmembrane domain. and a coiled-coil domain, and these tandem xylose and glucuronic acid transferase domains. These glycosyltransferase domains are like classical GTA domains, which means they chelate a manganese cofactor with a double aspartate motif, and they use a UDP sugar as a substrate. And their architecture is Rossmann-like, which means they have an alpha-beta-alpha sandwich. So if you flatten the secondary structure out of large one, you will get this beta sheet in between two alpha helical red pieces, I guess. And this architecture just repeats itself for the glucuronic acid transferase domain as well. AlphaFold, here is the AlphaFold monomeric structure. You can see it doesn't quite know what to do with the transmembrane domain or the coiled-coil domain, but it essentially gets the globular domain folding correct. So one of the things we wanted to optimize for large one was its pH that we were going to vitrify the grids at. So large one functions in the Golgi, and you can see that here in this anion exchange chromatogram, we've added the 4-methyl-umbelliferone substrate as well as UDP xylose and UDP glucuronic acid. So it can make a full chain of matriglycan. And what you see here in this first peak is the reagent, and then each of these peaks corresponds to one set of one set of xylose and glucuronic acid added to the fluorescent substrate. So you can see at pH 7, the chain kind of only extends to 14 sugar units, but at pH 6.5, it extends to 26 sugar units. So we thought, 6.6 is a good pH for the enzyme to be active, but usually we don't want enzymes moving around too much for structural analyses. You'll get all different shapes, but of course cryo-EM can deal with that. So we also did some thermal denaturation assays at 2 different pHs, and you can see at pH 6.6, the melting temperature is marginally better than at pH 7.4. So we went with pH 6.6 for grid freezing. One of the other things that we wanted to check out was what is the solution molecular weight of large one. So large one has a coiled-coil domain, which means it's going to form some sort of oligomer, and we wanted to know what that was. The theoretical molecular monomeric weight of large one is about 89 kilodaltons. So if large one was a monomer, we would expect like 90-ish kilodaltons on size exclusion chromatography followed by multi-angle laser light scattering. So this mold part gives you the molecular weight of a protein independent of its elution volume. So the data that you're seeing here is elution from a size exclusion column. The continuous line is the absorbance at 280. And on the secondary axis is the solution molecular weight. And you can see it's a little bit polydispersed, but not too bad. And the average molecular weight is about 220. So you can imagine 90 times 2 is about 180. It's a Golgi enzyme, so it's likely glycosylated. And so from this data, we think large one is probably a dimer that is glycosylated. And I'll say that this data was collected at BioCat Beamline at the Advanced Photon Source in Chicago. So to confirm that it was in fact some sort of glycosylation that was going on here, we digested large one with PNGase F. We know from mass spectrometry data that there are some N-glycans hanging around. So in the dotted cyan line here is the PNGase F digested large one. And here you can see the light scattering gives you a molecular weight of roughly 180 kilodaltons, which is almost exactly double of 90 kilodaltons. So that tells you that probably the higher molecular weight, which is not an integer here, which is about 2.2, is because of the glycosylation. And once we remove the glycosylation, you can see that this light scattering curve across the peak is a lot flatter, which tells you that the polydispersity here, the multiple molecular weights, is probably due to heterogeneous glycosylation. So we kind of keep that in our back pocket. One of the other things we wanted to do was to check out this protein called large 2, which is a gene duplicate of large 1. And it is missing the coiled-coil domain. So this protein allowed us to distinguish whether the coiled-coil domain was contributing to the dimerization. And what we saw was large 2 also formed dimers independent of the coiled-coil domain, which suggests that the xylose and glucuronic acid transferase domains were sufficient for dimerization. And so at Argonne National Lab, the main reason we went to Argonne National Lab was they have a sac steam line. And so we can get a rough idea of what our large 1 looks like. So here's the molecular envelope of large 1. And its Dmax is about 180 angstrom. Its Rg is about 46, 47. And this work wouldn't have been possible without Dr. Zhen Zhu here at University of Iowa, and Jesse Hopkins and Maxwell Watkins, who work on site. So when we do cryo-EM, kind of maybe this is what we're looking for. These are the dimensions that we're looking for. At least we know what to look for on the grid. So one of the other things we can say from the molecular envelope is that large 1 forms parallel dimers, and in the Golgi apparatus, large 1 is likely forming an orientation where the N-terminus is in the cytosol. There's a transmembrane domain here, coiled-coil domains here, and there's a globular catalytic head, which encounters dystroglycan en route to the plasma membrane and then polymerizes matriglycan as it passes through. So then we went ahead and optimized some vitrification conditions. We used a vitrobot to vitrify grids. I will say initially we used copper 1213 grids, and the protein kind of aggregated around the periphery of the hole, but we still managed to get a 4 angstrom structure. Then we switched to ultrafoil grids. So this is kind of in the middle of the pandemic, and there was a shortage of pretty much everything. And so I told the company, just give me whatever you have. It doesn't really matter right now. And so they had ultrafoil 200 mesh grids. And I didn't optimize any further than looking at these 2 grids. I was just lucky, I guess, that my protein's a bit of a brick, and it kind of sustains itself on different media and different experiments, and it lives at 37 for a week happily. Nothing happens to it. The grids were made at a concentration of 0.2 to 0.4 mg per mil. 0.4 mg per mil honestly was a little bit crowded. 0.2 mg per mil is better. I'll say earlier in 2018, I did try to vitrify these grids in pH 7.4. So remember, pH 7.4 wasn't so great for enzyme activity or thermal stability, but actually all the sec-mol stacks data are collected at pH 7.4, and it looks great. The protein can go up to 10 mg per mil in pH 7.4, but it doesn't seem to freeze well in our hands. So in 2020, 2021, we refroze some grids in pH 6.6 with 150 millimolar salt and 2 millimolar manganese, which is essential, it turns out. We did some microscale thermophoresis assays where we titrated in manganese, and you can see that there's a binding, whereas magnesium and calcium really don't have a binding curve associated with it. So the manganese, it turns out, is labile and needs to be added back into large 1 in order for activity. So the vitrification conditions also were 94% humidity. At 24 degrees, this protein doesn't seem to like ice. There's other data that suggests from thermal denaturation that we shouldn't put the protein on ice. And of course, it's a soluble protein, so I didn't use any detergent. So initially, the grids actually were screened at University of Utah. And then we sent the grids to PNCC, and Omar, who introduced me, was the SPOC who collected pretty much all my data. So we collected about 5K micrographs on a Titan Krios at super resolution. We collected 50 frames in each of the movies, and the electron dose was 50 electrons per angstrom squared. We did not use an energy filter. I think it was just easier to get on a microscope without an energy filter, so whatever. Beggars can't be choosers. And Omar collected it using a program called cryo-EM, and we also analyzed it on the fly using cryo-Spark Live. Oh, I should say, like, the 2D class averages already looked fantastic as we were collecting the data, so I was pretty happy with that. The data itself looked really promising, and so it set me up for learning how to process the cryo-EM data pretty well. So at least 3 people have processed cryo-EM data for large 1. So Nick Schnicker and I, we independently processed our data set at the University of Iowa, and then Michael Katz from Ron Diskin's lab in Israel also processed a set of their data. So we're pretty sure this is the structure of large 1. So Nick and Michael Katz appear to have processed the data using conventional methods. So I'll say like normal 2D classification, and then ab initio reconstruction, and then non-uniform refinement. Because large 1 is a dimer, we both did C1 reconstruction and C2, just in case there was asymmetry there. So this is Nick's reconstruction of large 1. Here you can see the 2 protomers of large 1 colored in different colors. And this is 90-degree rotation. The glucuronic acid transferase domain is here on top, and the xylose transferase is here on the bottom. And this is a local resolution map of large 1. And it fits our model that this is what it looks like, the globular catalytic head. We could not see the coiled-coil domain, and you can see in some of the 2D class averages, there's this funny little density here. We believe that's the coiled-coil domain flopping around, but we can't be 100% sure. You can see here also there's like a little density here. I'll say that my workflow was slightly different. I also did several rounds of 2D classification and generated an ab initio model. Then I low pass filtered it and generated 3D templates. And I then went ahead and undertook a no 2D workflow. And so the advantages of a no 2D workflow is that the particles or the 2D classes that you think are looking like your particles, that is biased by what you think your particles ought to look like. And notice that, so this is from just 2D classification. Notice that none of these have a top or bottom view of large 1. And so when you repick the particles automatically based using a template from a 3D volume, and you don't bias it in any way, it's possible to get a much higher resolution structure. So here is the GSFSC for all 3 independently constructed large 1 volumes. So this is a GSFSC from Nick's. These are the GSFSCs from the group in Israel. The group in Israel, actually, they had 1 construct where they kept the coiled-coil domain and another construct where they had chopped it off. And what you can see is the one where they kept the coiled-coil domain, it forms a parallel dimer, but the one where they chopped it off, it forms an anti-parallel dimer. And this funny little artifact dip that is present in all three of our C1 reconstructions. Here, if you remove the coiled-coil domain, it ameliorates the dip. So what you can see with the no 2D flow is generally it will give you a higher resolution structure. I actually went back and did a 2D classification on all of the particles that were used in the no 2D workflow. And some of them ended up looking like noise that I would not have picked. So that kind of tells me maybe it is incorporating noise, or I don't know the quality of the particles it has incorporated to get what seems to be a higher resolution structure. So here I am showing you the change. Click on this. Don't know if I can click on this. Oh, shoot.
Usually you have to go out of the laser pointer back to the cursor.
How do I? Oh, yeah, got it. So here is the deep EM enhanced volume of the large one dimer with the cartoon model of each of the monomers from AlphaFold modeled into it. I use Phoenix Real Space Refine to just refine it roughly into here, but Mick used Coot to actually go through each of the residues and correctly model in each of the residues. So what you can see here is there is definite orientation bias. So so even though I can make the conclusions that I need from this structure, I cannot, I have missing density in several places. So for example, I have missing density right here. And this is true in all of both Nick and my C1 reconstructions, but as soon as you do it in C2, this density fills out. I do believe this structure is asymmetric for biological reasons. And so here I've just overlaid the cartoon on to orient you on the different domains. So the xylose transferase domain is down the bottom and the glucuronic acid transferase is at the top. And if you increase the contour level, you can kind of see where the coil comes out. So the major conclusion from this is that the active sites on alternate protomers face opposite directions. So that means the xylose active site, xylose transferase active site on one protomer faces the same direction as the glucuronic acid transferase site on the other protomer. And the GTA folds are really well known. There's nothing novel about them. The novel part is when they occur in tandem and their orientation with respect to each other. So this kind of tells you that matriglycan is polymerized by alternating between these two active sites rather than taking the xylose from this active site and then going around the globular catalytic head to add a glucuronic acid from the other active site on the same protomer. That seems fairly inefficient. So this kind of pointed towards a processive mechanism, and this is where the hypothesis generation from the cryo-EM reconstruction comes in. We wanted to know whether large one polymerizes matriglycan processively, and if it does polymerize matriglycan processively, this has implications on the length of matriglycan that is polymerized in disease states. So if you interfere with the processivity of large one, you might end up with shorter matriglycan and you might end up getting various diseases related to muscular dystrophy. So Processive polymerization generally results in minimal intermediates. So you'll have a starting material here and then the finishing material here. And it usually gives you discrete product lengths. Whereas distributive polymerization, that's when the enzyme substrate complex dissociates between catalytic grounds. You'll be able to see intermediate products. So your gel will look like a bit of a smear. You'll also get a Poisson distribution of products. So remember the anion exchange chromatogram that I showed you earlier with 4-methyl-umbelliferone and matriglycan polymerized on there. So here you can see the products have a Poisson distribution, and this definitely is a distributive mechanism with this particular substrate. But we want to know what it looks like with the native substrate. So in order to do that, what we designed was a recombinant soluble dystroglycan that had the same conformation as in the Golgi. So one of the things about dystroglycan is that it's a pre-pro protein. It's translated as a single polypeptide, and it gets auto-cleaved into alpha and beta chains, which remain associated. And then once it travels to the Golgi, there's a bunch of post-translational modifications that occur in the ER, travels to the Golgi, gets a couple more post-translational modifications before matriglycan can be synthesized. When it gets transported to the plasma membrane, it actually loses this globular N-terminal domain called DGN. It has a furin cleavage site here, so furin convertase clips the protein. But in the Golgi, when large one encounters dystroglycan, it needs this DGN moiety. So what we did was we mutated the furin cleavage site into an alanine, and we chopped off the membrane portion, so we have this secreted soluble form of dystroglycan. And this is what we used as a substrate for matriglycan polymerization assays. Now, the other thing that large one allows us to do is to make active site mutants of large one. So here, the xylose transferase site is mutated, and here in this construct, the glucuronic acid transferase site is mutated. And when you add these two things together, we can still polymerize matriglycan, but in a purely distributive manner. And so we will know what the product looks like when we add these two things together. So this is DXD1 and DXD3, but we don't know what the wild type would look like, right? So when we compare the two products, the purely distributively synthesized product and the product made by just wild type protein, we might be able to distinguish the mechanism. So here I'm just showing you another anion exchange chromatogram. Here is wild type large one making matriglycan. You can see the individual mutants, the XylT and the glucuronic acid transferase mutants don't polymerize matriglycan. And here in purple is the matriglycan polymerized by both DXD1 and DXD3 mixed together. And this is the Western blot of matriglycan polymerized on the recombinant dystroglycan substrate that we engineered. So here you can see DXD1 and DXD3 polymerizes matriglycan just fine. And this is the product from the wild type protein, which looks different from the purely distributively polymerized protein. So here in the wild type protein, you have a very discrete band followed by a smear. And that discrete band is missing in the distributively polymerized matriglycan. This is our positive control. So this is matriglycan synthesized on dystroglycan using endogenous large one. So HEC293 cells have large one expressed, and this is what they polymerize on the recombinant dystroglycan. So from the cryo-EM structure, what we can say is that large one proteins are dimeric. They use orthogonal active sites on alternate protomers to processively polymerize matriglycan, specifically on dystroglycan, but not the synthetic substrate. And somehow the large one dystroglycan enzyme substrate complex knows what length of matriglycan to polymerize. And we know that there's some sort of molecular ruler in there. So in the future, we'd like to know the structure of the enzyme substrate complex and how interfering with processivity results in short matriglycan. We'd also like to explore, using structural and biochemical techniques, what factors affect the processivity of large ones. And that's the end of my talk. I'd really like to thank the Campbell Laboratory. This is us in a recent picture. I'd like to thank Sandeep Chowdhury, who helped me, who taught me how to process the data, both with the 2D reconstruction and the 3D no 2D workflow. Nick and Jen did a great job interfacing with both the facilities, both PNCC and Argonne National Lab. And I'd like to thank the High Performance Computing Center at the University of Iowa. Before I got my high performing workstation, I used the clusters here at the HPC. Externally, I would like to thank people at Argonne National Lab, formerly Srinivas Chakravarti, who used to work there, Jesse Hopkins, Maxwell Watkins, Dr. Dave Tim from the University of Utah, and of course, TNCC. Without that facility, I think this work would have been impossible. And specifically Omar, who collected my data for me at TNCC. Yeah, questions?
So thank you, Soumya. That was a really interesting talk. We've got a couple of questions here to get through. I know you mentioned using AlphaFold to predict some of these models. Could you tell us a little bit more about what models you actually used it to predict, and specifically whether you looked at the entire complex with AlphaFold to see if you had any prediction of where the coiled-coil domain would lie?
I looked at the monomer of large ones because I didn't want to bias it. So I just modeled 2 of the monomers in. There are other ways that I did kind of validate the interface. I looked at the surface conservation using Conserve to check that, you know, the residues at the interface were conserved. I did not specifically look at the coiled-coil because it wasn't resolved in the cryo-EM reconstruction. Let's see. Yeah, I didn't have a chance to kind of model that with the data that we had.
Yeah, that's great. So another question, how did template picking with those 2D classes that you selected progress? How did that work out for you?
Yeah, the templates produced better and better resolution reconstruction, and it made more and more sense as I went through the cycles of template picking. Yeah, I did
so
about 3 steps. Yeah.
Sorry to interrupt you.
And then,
so you mentioned that you know, the matriglycan is kind of passed from one domain to the next. Is there any indication how it might be held and actually transferred between these alternating sites?
Yeah, so how it's held, I'll answer that one first. So we know that the globular domain here, DGN, is absolutely necessary to make that punctate band. And so we think it's actually a protein-protein interface that is holding the enzyme substrate complex in a complex. And the second part of the question, how does it transfer? Gosh, I have a suspicion as to how it transfers, but I don't know. I can't really answer that question. we did do some 3D variability analysis, and there is a little bit of a rocking motion between the protomers. And there definitely seems to be some sort of communication between the xylose and glucuronic acid transferase domains. So if you happen to mutate a residue in the glucuronic acid transferase domain, you can lower the activity of the xylose transferase domain. But I haven't done enough of the analysis to understand how the reducing end wiggles between the two, whether it's just stochastic or is there like a channeling mechanism in the protein itself.
All right. And so we've got a question about the gels and the westerns that you were showing. Could you possibly speculate on the identity of the discrete band seen when dystroglycan is treated with wild type large 1? And also whether you would expect the product of a processive bidomain GT. I'm not sure what that stands for, but would you expect that to migrate?
Transferase, yeah.
Okay. Would you expect that to migrate at the top of the gel, or is it penetrating the gel because it has a strong negative charge?
Okay, the identity of the discrete band. So because we have purified the recombinant construct, and we actually purify it using nickel NTA resin, and then we do an anion exchange to make sure we get the starting material without matriglycan. The identity of the discrete band, I would say, is it has some length of matriglycan on it. We actually don't know the length of the polymer. That is one thing that is up in the air. We've been trying to solve that for a long time. So it's the recombinant dystroglycan with a certain length distribution of matriglycan, which we don't know what it is. And whether the product should penetrate the gel, yes, it does penetrate the gel. Even if you extract the protein from native tissues or patient samples, the alpha dystroglycan, because it is not covalently linked to the beta dystroglycan, it's actually small enough to go through the pores of a polyacrylamide gel. And yes, you're right. The matriglycan itself is negatively charged, so every glucuronic acid on the protein adds a negative charge to it.
So maybe a naive follow-up, could mass spec help you pin down the length?
Yeah, so we tried that, and I'm not sure if I'm allowed to say. We tried it, and the results aren't out yet. So what does that tell you?
All right, so we'll just hold on to that thought.
If
so
I
that's
can
our last question.
Actually, if I can chime in with a question, because I can unmute myself. So, and maybe I missed this. So are you using the glycosylated or deglycosylated protein for making your grids?
Yeah, so I'm actually using the glycosylated protein for making my grids. So in my model, I did put in the N-glycans, but it doesn't have density for the N-glycans in the reconstruction. So I'm guessing the N-glycans are a bit wiggly and they've been averaged out.
And then another question. So your sort of half dead constructs that you're using for activity assays, do you have plans to put those on grids, or have you put those on grids? I'm wondering if those in combination with some sort of substrate might get you an interesting structure.
Yeah, I actually put them on grids last week. So we'll see where that gets to.
Fantastic. I'll be keeping my eyes open.
If I could add a question, which the panelists aren't allowed to put in the question and answer. I just want to get an idea of which end of the polymerized matriglycan is stuck onto the dystroglycan. Is it the end that was the last synthesized one, or is it the end, the first ones that were made that get eventually stuck onto the dystroglycan?
Yeah, absolutely. So I will say two things. One, I don't think large one is capable of binding the matriglycan product in a very tight manner. And the reason it won't do that is because enzymes generally don't like to bind their products, and this particular enzyme will, it needs to oscillate between the active sites. So it needs to be fairly mobile in order to do that. The sugar that is actually binding it is this guy. So we have previous data that's already been published that this core phosphorylated mannose can bind large one with a affinity of 11 micromolar. So that's using a NMR titration that we know. So it's this entity, DGN here, that appears to bind large one, and somehow this sugar appears to bind large one. And the patient data that I showed you earlier with the very short matriglycan, that patient lacks the enzyme that puts on this phosphate. And so somehow there is this phosphorylated trisaccharide that activates large one in order to polymerize full-length matriglycan. So yeah, in order to answer your question, it would bind this end, but not necessarily matriglycan. It binds this stem core. Yeah.
All right. Thank you so much, Soumya. That was absolutely fantastic. I had so many questions while you were talking, and then you answered all of them along the way. That was so thorough and fantastic, and you hit all the details. It was great. And I hope everyone else here found that very useful. And we hope all of you in our audience good success in your own cryo-EM endeavors. The national centers are here to help you do your science. Hopefully you come find us if you're in Portland. Otherwise, reach out to us at any point. We're here to help you, and hopefully we see some of you here again next month for our next talk. Look out for those advertisements, and we will see you then. Thank you very much.